I'd like to introduce you all to Dr. Ken White, who's doing the last of our summer research lectures. It's my great privilege to present Ken White. Thank you very much for coming.
Thank you for inviting me, Dan. I'm from the Molecular Systems for Health Research Group based at the School of Human Sciences, and one area of research I'm particularly interested in is harnessing phytochemistry to combat diabetes. So the title of the talk is Phyto-solutions for the treatment of diabetes, harnessing ancient remedies for an ancient problem. So I shall talk about the sort of historical viewpoint of diabetes very briefly. I'll talk about the incidence of diabetes. I'll give some description of the disease and its pathology. Also talk about current treatments because, of course, it is being treated reasonably successfully already. So why should there be a need for new types of therapy? And then that'll provide the background to talking about herbal approaches to treatment and some of the work that we're doing at LMU here. And finally, acknowledging the hard work of the people who have done most of the lab work. So diabetes has been known about and written about for quite a long time. So one early description was in the 1st century of the Common Era by a Greek physician called Aretaeus in Cappadocia, which is actually in central Turkey. And he described some of the key symptoms of diabetes, that is to say excessive urination, and also he noted that the sufferers of the disease had a short, painful and morbid lifetime. Quality of life was very, very low indeed. Later on, Avicenna of Persia, now Iran, gave a detailed description of diabetes, including symptoms, again, of over-urination, but also the fact that the urine was sweet. And generally there was a wasting of the body and sometimes there was gangrene. But he was also fairly observant and identified 2 types of diabetes, which we now call type 1 diabetes and type 2 diabetes. In those days there were treatments available, local herbal treatments available, including the use of trigonella, or fenugreek as we now call it, and also lupine and zedoary seeds were also used as part of a remedy for diabetes. Of course, we're all aware that diabetes has become endemic. It's a global problem, and this is data from the International Diabetes Federation published in 2013, and these numbers increase year by year. So these data are now 2 years old, but around the world it is thought or estimated that there are about 382 million people living with diabetes, and about half of those will not be diagnosed. And we can see from this global map that the incidence isn't just really in Europe and North America, but it's actually across the whole globe. So 138 million in the Western Pacific, 72 million in Southeast Asia, 35 million in Africa, and 20 million in South Africa, 24 million in South America. So it really is a global phenomenon, and it's endemic. And if we look at individual countries, we come up with some perhaps surprising observations. In terms of actual numbers of sufferers from diabetes, China has by far the largest number, 98.4 million in 2013. Next is India with 65 million. And if we look at that top 10, we can see the US, Germany, Russia, and the number 10 is Japan with 7.2 million sufferers. If we convert those figures into percentage of population, we come up with a different story. We see that in the Polynesian islands, in the Pacific, the prevalence of diabetes is extremely high, 24 to 37%. And in Saudi Arabia, Kuwait and Qatar, it's 23%, and so on. In the UK, it's about 5%, compared with about 8% in Germany and Russia, and I think it's about 7% in Japan. So as a nation in the global league, so to speak, we're not doing too badly, but it's still 5% of the population is a huge number of people. And of course that number is going up year by year. 3 types of diabetes are now recognized. Type 1 diabetes, which is caused by a failure to produce insulin. Type 2 diabetes, which is due to insulin resistance. And then there's gestational diabetes, which occurs sometimes during pregnancy, where you have a very high blood glucose level. So diabetes develops when the mechanisms by which we control the amount of glucose circulating in our blood don't work properly. There is no longer homeostasis of glucose in the body, no longer systemic glucose homeostasis. There's a breakdown of that. And that means that in the blood circulation, the concentration of glucose is higher than it should be, normally about 8 to 11 millimolar instead of the normal upper limit of about 5 millimolar. If there's too little glucose, the concentration is less than 5 millimolar. So having too much glucose is bad news, having too little is also bad news. The level of glucose is controlled by a key hormone called insulin. I'm sure you've all heard of insulin and are vaguely aware of what it does. It regulates the amount of glucose that is taken up by tissues around the body, in particular by muscle and by fat tissue, but it also regulates production of glucose by the liver, as is illustrated in this slide. Now, the key organ which produces insulin is the pancreas, and so the pancreas itself is like a glucose sensor. It can release insulin in response to increases in glucose that occur in the blood after you've eaten a meal, for example. But if that doesn't work, if, for example, you have too much loss of glucose from the blood, we have hypoglycemia. And when that happens, you tend to lose consciousness. So this is a very important symptom of diabetes, that if you don't have enough glucose circulating in the blood, you lose consciousness. And the reason for that is that about 30% of the glucose in our blood supply goes to the brain. The brain consumes a lot of sugar, needs a lot of sugar to remain active. And when the sugar levels fall, in the short term, you'll lose consciousness. And of course, if you're not aware that you're a diabetic and you start losing consciousness and you're doing something very important like driving a car, that can be a big problem. On the other hand, though, if you have too much glucose, the consequences aren't immediately obvious. But over a long period of time, chemical damage tends to build up. And glucose itself, from a chemical point of view, is an aldehyde. And that means it can react rather non-specifically with proteins and lipids on cell surfaces, and that can affect the functioning of tissues. Of course, it doesn't do this very, very quickly. This is a build-up over a long period of time. But taking advantage of that reaction, we can use a blood marker for diabetes, which is glycated hemoglobin. HbA1c is the key blood marker for measuring the extent of your diabetes. This slide illustrates in a rather complicated way some of the consequences of having glycated proteins, that basically cell function is impaired. It doesn't stop altogether, it doesn't cause cells to die, but it means that tissues become less flexible, that blood supply at the periphery of your limbs becomes less available, and so in fact the supply may be cut off completely, eventually causing gangrene to develop. Normally the symptoms are picked up well before that stage, but sometimes in extreme cases people do have limbs amputated because of diabetes. So the pathology of diabetes includes damage to the heart, which is the most common cause of death in diabetes, but also very commonly damage to kidneys, and also eye problems as well. Retinopathy is a symptom of diabetes, and indeed, if you go to the opticians regularly, as they like to tell you, that they can pick up early signs of diabetes by examining the retina very closely. And nerve damage is another outcome of diabetes. When we look at people to see whether they are becoming diabetic, we monitor their blood glucose very carefully. We give them perhaps a sample of glucose and see how quickly that glucose is removed from the blood. So this slide illustrates some of the key figures that are looked at in terms of blood glucose consumption. So that if you look at the fasting plasma glucose after a normal dose of glucose, then you should be able to bring the concentration of blood glucose back down to about 6 to 7 millimolar fairly quickly. If that doesn't happen, that's an indication that perhaps you have diabetes. And there are different ranges according to the type of diabetes that you have. Another way of looking at diabetes is to look at the what's called insulin sensitivity. that in the case of type 2 diabetes, you produce insulin. It's not the lack of insulin that's a problem. It's the fact that the tissues become unresponsive to insulin. They lose what's called insulin sensitivity. And insulin sensitivity can be measured. And if you're within a range on this curve between A and B, then you have what's called normal glucose tolerance. But if you're outside the range, if you go from B to C, then you have incipient diabetes forming. And if you go as far as D, then you're really in the range of a type 2 diabetic. So it's losing this sensitivity to insulin that's one of the key problems with type 2 diabetes. Why does that happen? We're not really quite clear about that, but it's a very clear sign of diabetes. So what does insulin actually do? Well, I've already said that insulin regulates the amount of glucose in the blood, and it does that by promoting glucose uptake into the muscle, promoting glucose uptake into fat, and also preventing glucose synthesis in the liver, amongst other things. It also affects fatty acid synthesis in the liver, and so it has a range of metabolic activities, but the main focus is its ability to regulate the amount of glucose in the blood by regulating the amount that goes up into the muscle or into adipose tissues, which are the main stores of glucose when we've had a meal. Treatment of diabetes depends on the type of diabetes that you have. So if you're a type 1 diabetic and you're not producing insulin, which is the characteristic of type 1 diabetes, then insulin is given as the treatment, that you have to inject yourself with insulin, or perhaps you have an infusion pump, or perhaps some other way of administering insulin. More modern approaches include the possibility of perhaps having a pancreas transplant, or perhaps even some sort of islet cell transplantation, or even stem cell therapy. So those are new types of way of treating type 1 diabetes, which are rather experimental at this stage. But the long-established method is to use insulin. But this won't help you with type 2 diabetes, because you're already producing insulin. Instead, you have to try and take some medicine which promotes glucose uptake by the tissues. And there are, in fact, a variety of drugs with a variety of mechanisms of actions which are used to treat type 2 diabetes. I'm not going to go through them all and describe them how they work all together, but I'll just give you a snapshot in this slide here, which shows various key tissues involved in diabetes. The liver on the top left, we have adipocytes center top, we have the muscle center right, and we have the pancreas down there bottom left. And depending on what the drug is, it has different mechanism of action. So, for example, biguanides, as they're called, and I'll come back to those in a minute, they prevent glucose release from the liver. They also promote glucose uptake by the muscle, for example. So there are various modes of action by different drugs. And of course, the response to the drugs depends very much on the patient, as we shall discuss in a minute. So these are well-established medicines, and in particular metformin, which I shall talk about more in a minute, which is an example of a biguanide. That's been used for over 50 years to treat type 2 diabetes. So it's a well-established therapy. But all medicines have some side effects in some people. I think that's more or less a truism in clinical pharmacology. And I think one of the reasons for that is that we are different. Obviously, we are different as individuals, but also genetically we are different. There's actually quite a big input of variants in the key genes which are involved in drug responses or drug metabolism. And this is called pharmacogenetics. And there's a huge field of pharmacogenetics or pharmacogenomics which deals with that aspect of clinical pharmacology. And I think for the same reason, some people have bad responses to medicines which work perfectly well in other people. So that's one key reason why we're on the lookout for new types of therapies. So in this list here, I've listed the main types of drugs used to treat diabetes, type 2 diabetes, and some of the side effects that they can induce, don't always induce, but can induce in some people. So for example, the sulfonylureas tend to cause hypoglycemia, that sometimes the glucose goes level goes down too much in some people. And of course, that makes them lose consciousness, which is very undesirable. In some people, the side effect is merely to make them put on weight, which is not good news, as we know that putting on weight predisposes people to develop heart disease and perhaps exacerbate the diabetes, and so on. So there's quite a long list there of side effects from the key types of medicine used to treat diabetes. If we focus on metformin, which I think is one of the most successful medicines that's probably in existence, because it's been used successfully on millions of people, tens of millions of people probably, over a period of 50 years. So it's a real success story. But nevertheless, in some people, it can have bad effects on the gastrointestinal tract. It can make people nauseous, give them diarrhea, and perhaps also they develop intolerance to lactose, so they can't eat dairy products. So that brings me on to the main theme of the talk, herbal approaches to treating diabetes. That we know over a period of many centuries, really, that herbs and plants have been traditionally used to treat diabetes. And that herbal formulations could provide an alternative to the modern medicines that I've talked about already. And one of the attractions of using herbs is that from a local point of view, that if you're in a country where perhaps the standard of living is not so high, or where people don't have high salaries, or they are poor, they don't have money to pay for expensive Western medicines. Therefore, if one can actually identify a local alternative and identify perhaps the key components of that local alternative, that may make give them an affordable treatment for that disease. And of course, there's always the attraction that there may be fewer side effects, although I think probably there will be side effects in some people for some types of herbal medicine. In addition, there's the prospect that we may discover new mechanisms of action, that there may be some way of modulating particular molecules that we don't quite know about at the moment that are waiting to be discovered using these new types of treatment. So there are potential roles then for introducing new types of phytochemical to treat diabetes, in particular berberine, fluoretine and wabane. They all have potential anti-diabetic activity, and berberine in particular has quite an extensive pharmacology that's been well worked out in animal studies and is beginning to be looked at in human trials as well. Metformin, which was, I've already discussed, has been extensively used for type 2 diabetes, is derived from a plant. And this is the story of metformin a little bit in summary, that this plant here, Galega officinalis, was recognized as being used successfully to treat diabetes as a herbal remedy. So that once you know that, the next step is to try to identify the active component from that plant. In the case of Galega officinalis, the active component is a chemical called galagine, which is the second structure on the list at the top. The top structure is guanidine, which is a very common metabolite in biochemistry, so that's very well known from normal metabolic biochemistry. But galagine is a derivative of that. It's actually it's two molecules of guanine joined together. And when that was investigated, purified and used in animal studies, it was found that it did work, but it actually had some toxic side effects. And so going on from that, it was decided to try to make some derivatives of galagine to see whether the side effects couldn't be modulated or removed, and also improve, in fact, the availability of the drug once you'd taken it orally. And to cut a long story short, metformin turned out to be the best out of all the derivatives that were tried. So metformin is a derivative then of galagine. It doesn't occur naturally, but it was discovered through studying the natural ingredient of this Galega officinalis plant. So from a phytochemical approach then, a very successful drug has been developed. So and I suppose in a way we want to try and repeat that success. And so the herbs that we've started to look at are all derived from histories then of treatment of diabetes. And I'm going to talk mainly about fenugreek. And this story starts in Iran through a collaboration with a colleague Reza Hairi, who we've collaborated with over quite a long time now. But we're also looking at other herbs, Stevia bordia as an Ayurvedic type of herb, Moringa peregrina, another herb that comes from Iran, and also bitter gourd, which actually you can buy in this country as a vegetable, is also potentially a promising anti-diabetic. Now, fenugreek, some of you may be familiar with fenugreek, and let me just show you a pack of fenugreek seeds that you can buy. You can actually buy these in supermarkets, or you can buy them in larger bulk from wholesale distributors. And in fact, my next door neighbour, who is an Indian lady, has said that she uses a lot. She uses it by the kilogram and uses it to make spices for her curry. So I think it's quite well known in this country in some circles. I must say I hadn't heard of it when I first came across it. But it has actually been studied for its anti-diabetic potential, and in particular, one key compound has been identified, which is apparently unique to this plant, and it's called 4-hydroxyisoleucine. So I shall talk quite a lot about this 4-hydroxyisoleucine and the work that we've done to study its anti-diabetic properties. So there's fenugreek, otherwise known as Trigonella Phonum graecum, and it's widely cultivated in the Mediterranean area, in the Middle East, in India and in China. So it has a long history in the use as a treatment for diabetes. And as I've already said, the seeds are used for cooking and for grinding up, and including in curries in particular. You can actually grow the seeds into seedlings and eat them, the shoots, rather like eating bean sprouts as well. The seeds are the major source of 4-hydroxyisoleucine, and there's the chemical structure at the bottom. You'll notice it's very similar to the naturally occurring amino acid isoleucine. There are 20 amino acids that make up our proteins. One of those is isoleucine. This is a derivative of isoleucine called 4-hydroxyisoleucine, and there's the 4-hydroxyl group there. Now, from a chemical point of view, this structure is a little bit it's fairly simple, but it has some complications in terms of its stereochemistry, in terms of the shape that it can have, because it has 3 carbon centers which have alternative ways of configuring the side chains. So in fact, you have 8 possibilities for the structure of that molecule. So out of those 8 possibilities, one of them is particularly pharmacologically active, and this is the 2S3R4S version of 4-hydroxyisoleucine. I won't refer to it as that, but just to point out that out of 8 possible structures, one of them has the key pharmacological activity. So there are the 8 possibilities. So one of those is the 2S3R4S, and I'll leave you to try and guess which one. In addition to the 2S3R4S, there is in fact the 2R3R4S version, which has about 10% of the pharmacological activity of the other isomer. But the most abundant isomer has most of the activity. In contrast, isoleucine exists as a single type of shape with a 2S3S conformation. So we've taken this 4-hydroxyisoleucine from fenugreek seeds, and we've used it in 2 types of animal model, which I'm going to describe now. One animal model was of type 2 diabetes. So we looked at the effect of fenugreek 4-hydroxyisoleucine on the liver function biomarkers in particular, and glucose levels in the type 2 diabetic model that we've used. And the type 2 diabetes is induced in rats by overfeeding them with fructose. So the fructose-fed rats are the ones which have symptoms of type 2 diabetes. And some data about the glucose homeostasis of those rats is shown here. That if you look at the level of glucose in the blood, you'll see that the control animals have about 160 or so milligrams per deciliter of glucose. And if you treat the control animals with 4-hydroxyisoleucine, that's the C4H bar there, there's no change. In other words, the 4-hydroxyisoleucine doesn't alter glucose levels when they're normal. It doesn't make you hypoglycemic, which is very important. If you looked at the fructose-fed model, the type 2 diabetic model, the F bar here, you'll see that the level of glucose circulating in the blood of those animals is much higher, almost about 280 milligrams per deciliter. But when you treat those animals with 4-hydroxyisoleucine, the levels come back down, not quite to the control normal levels, but a lot better than 280, roughly about 180 milligrams per deciliter. So that tells us that the 4-hydroxyisoleucine has the ability to lower blood glucose and therefore has anti-diabetic properties in this model. And we also looked at the effect of the amino acid on the function of the liver, because one possibility of feeding large amounts of this amino acid is inducing rather toxic reactions or side effects that you don't want to occur. So as part of screening those side effects, we looked at the effect on liver function. And the way that you do that is to assess this amount of circulating enzymes in the blood, which are released from the liver. There's also a little bit of turnover of liver cells and release of these liver-specific proteins, if you like, into the blood. And we can see once again that in control animals and control animals treated with 4-hydroxyisoleucine, that there's no change, that in the fructose-fed animals there's an increase in liver damage induced by the diabetic state, but that increase is actually ameliorated, is actually reversed by treatment of 4-hydroxyisoleucine. So in other words, it's good news that the 4-hydroxyisoleucine actually reduces liver damage in these diabetic animals. Another part of our research is actually to try and find ways of preparing useful amounts of 4-hydroxyisoleucine. The problem with this type of study is actually having enough 4-hydroxyisoleucine available to use on animal studies. You need perhaps a gram of material, which may not sound very much, but when you're isolating about 0.6 of a gram from 100 grams of seeds, it's hard work to do that on a regular basis. So one of the ways in which we try to improve the yield of obtaining 4-hydroxyisoleucine is to actually, first of all, we looked at the amount of amino acid in the seeds versus the seedlings, but also we fed the seeds with extra isoleucine, because isoleucine gets converted to the 4-hydroxyisoleucine, and by doing that we can actually increase the yield of 4-hydroxyisoleucine about 6 times. So that's been a useful little side piece of work to help us in our studies further on. And the next stage then was to look at the effect of 4-hydroxyisoleucine in a model of type 1 diabetes. And type 1 diabetes can be induced in rats by feeding them with an antibiotic called streptozotocin, or STZ for short. And it turns out, this was just a chance observation, but the STZ selectively is toxic to pancreatic cells. For some reason, pancreatic cells are very sensitive to this antibiotic. And so they are killed effectively, and so the production of insulin is decreased dramatically. And that's, we show that later on in the data, but when you do that, and this is a standard way of inducing type 1 diabetes in rodents in particular, when you do that, you can see that in the control animals on the left, the level of glucose in this case is a little bit low, about 100 milligrams per deciliter. But in the diabetic animals, it's shot up about 5 times to well over 500 milligrams per deciliter. And then when you treat them with 4-hydroxyisoleucine, the levels come down, not nearly as much as control, but they are improved quite significantly. And the level of insulin was quite important to measure the effect on of the STZ on the level of insulin. After all, if the insulin is still being secreted, or is secreted, or in fact its secretion is increased by 4-hydroxyisoleucine, then that has a profound implications about the mechanism of action. But in fact, it turns out that the level of insulin is about half, or perhaps slightly under half, in the diabetic rats compared with controls, and the 4-hydroxyisoleucine doesn't make any difference to the amount of insulin that's released. So this is a very important conclusion, that the effect of the 4-hydroxyisoleucine is not due to increased production of insulin. It's some other effect on the peripheral tissues, presumably. And we did some basic measurement of food intake, water intake, body weight, to make sure that the animals were behaving according to the usual models of diabetes. so that a diabetic animal will actually drink a lot more, will eat a lot more, and those diabetic animals were treated with 4-hydroxyisoleucine. The water intake after 4 weeks fell off a little bit. It wasn't quite, well, it wasn't down to normal at all, but it's still a trend in the right direction, and they certainly ate less significantly than the diabetic animals, so that's all encouraging as well. And also encouraging was the profile of lipids in the diabetic animals, which was improved by treatment with 4-hydroxyisoleucine. That the normal profile of lipids, of cholesterol, low-density lipoprotein, high-density lipoprotein, cholesterol and triglyceride, that the profile of those which are indicative of metabolic disease in the case of the diabetic animals, they are reversed to some degree, either back to control levels in the case of LDL, or reversed a little bit in the case of HDL, reversed a little bit in the case of total cholesterol, and reversed substantially in the case of triglycerides. So all these are improvements on the lipid profile caused by the 4-hydroxyisoleucine treatment. And finally, the level of uric acid, which is an indication of kidney malfunction, was restored to normal by treatment with 4-hydroxyisoleucine. So all in all then, quite a successful improvement of the symptoms of diabetes in this animal model. So in conclusion then, 4-hydroxyisoleucine is effective both as an anti-diabetic treatment both in type 1 and type 2 diabetes. It is a safe compound, it's well tolerated, it has no side effects that have been discovered so far, although of course we need to do a lot more work to establish that, but others have also used this compound in isolation and nothing has been reported yet. Intriguingly, the mechanism of action has not been worked out. This is much more difficult to work out. Which tissue is it particularly working on? What types of proteins or enzymes are modulated by this very small molecule? That's a question of research which really hasn't found its focus yet. So current work then is that we're studying the mechanism of action of 4-hydroxyisoleucine by using it on cultured cells, cells representing, for example, skeletal muscle or liver or adipocytes, and trying to find out what key molecule might be regulated by the 4-hydroxyisoleucine. We've done quite a lot of work on beta pancreatic cells, and we have quite a lot of evidence to suggest that there may be a tendency for 4-hydroxyisoleucine to uncouple oxidative phosphorylation. That means it makes the mitochondria, which makes ATP, it makes them a little bit inefficient in their production of ATP, so that instead of producing ATP, the glucose in fact produces heat. And some of you may be aware that some of the slimming treatments which are available on the web, and tragically this has happened not so long ago, that one uncoupler of oxidative phosphorylation called 2,4-dinitrophenol, which is available freely on the web, was actually used by a young lady as a slimming tool, and she actually poisoned herself by taking it too much. So these compounds are very potent. They're mild poisons of mitochondria. But if that poisoning effect or that inhibiting effect is very, very mild, then it can be well tolerated and maybe one way in which the 4-hydroxyisoleucine works. So we're currently investigating that. We're also interested, as I said at the beginning, to try and look at active components from other herbs, in particular Ayurvedic herbs, the Stevia rebaudiana, and so we're isolating and characterizing active phytochemicals from that plant. Moringa peregrina, similarly, and finally the bitter gourd, which you can buy as a vegetable, usually in shops from the Middle East or from the Mediterranean, in particular, for example, Turkish shops. You can buy this. Sometimes it's called bitter melon. If you ever eat it, it's supposed to be very good for you, and it's actually a delicacy in Japan and China and elsewhere. It does taste very, very bitter, but it's supposed to be very good for you. But it includes in it an anti-diabetic peptide, which has not been well characterized. It's a very small protein of about 100 amino acids, and we're quite interested in isolating that protein, sequencing it, and trying to find out how it works. And finally, I'd like to acknowledge the collaboration and work of a group of people over the years. All of this was really started by a visit from Dr. Reza Haeri from Iran, who's now at the Tehran University of Medical Sciences. He introduced us to 4-hydroxyisoleucine, and he persuaded Hamid Kalakbari to do his PhD in this area, and Davud Rustai and Kiran Kundanuri are also continuing in this area, or they're finishing off now. And Dr. Lippy Das, she's currently working with me to try and work out the mechanism of action for 4-hydroxyisoleucine. And John Morgan at the LMU Science Centre has helped with extracting 4-hydroxyisoleucine from fenugreek seeds. And thank you very much for your attention.